Hello, BISC130. This is the beginning of recorded lecture 3.2. Continuing on with chapter 10, cell reproduction. In the previous recorded lecture, we had talked about the eukaryotic cell cycle and all these different proteins involved in regulating this cycle, and pointed out that there are so many checkpoints involved here. It's highly regulated. Cells are only supposed to divide when they need to divide and when they are able to divide correctly. This is how things are supposed to work. However, nothing is perfect. Nothing ever works the way it's supposed to 100% of the time. It is possible to get cells that are not properly regulated, that divide when they are not supposed to divide. This leads us to the next topic, cancer. So we could define cancer as the uncontrolled cell growth, which causes harm. And it's not difficult to cause harm. It's pretty easy for uncontrollably growing cells to cause harm to the nearby tissues, nearby organs. Usually uncontrolled cell growth leads to harm to the surrounding stuff. So cancer, this uncontrolled harmful cell growth, is caused by failures of all the stuff that we talked about at the end of the previous recorded lecture. Cancer is caused by faulty cell cycle control proteins. Any of the proteins involved in these checkpoints, these regulation mechanisms, not doing what they are supposed to do. So, okay, what causes these proteins to fail? Well, we know that proteins can become denatured through changes in pH and temperature, but actually every single one of these cell cycle control proteins, there are many, many, many copies of these in any given cell. You don't get dysregulation by, a few denatured proteins. What causes bad cell cycle control proteins is mutations in the DNA sequence. Every single protein we talk about in this entire course comes from a gene. It has to have a blueprint. There's a gene for cyclin A, there's a gene for CDK2, there's a gene for, there's cyclin A down here again, just trying to pull out random things. There's a gene for DUX4, there's a gene for all these things. If the blueprint for cyclin A gets screwed up, every single cyclin A protein that the cell makes is gonna be screwed up. So these faulty cell cycle control proteins are caused by mutations in their genes, mutations in the DNA sequence. Okay, let's continue down this. What causes mutations in the DNA sequence? Well, mutations in the DNA sequence are caused by unrepaired DNA damage. Our damage is being DNA'd. Our DNA is being damaged constantly by various things. We'll talk more about mechanisms of this in a different chapter, but we have ways to fix this damage. If our DNA damage is not fixed, that can lead to a mutation. That can lead to these things not doing what they're supposed to do. There are a couple of terms here: proto-oncogenes and oncogenes. You may recognize the prefix onco. Onco means cancer. So a medical doctor specializing in cancer is an oncologist. A medical facility specialized for cancer patients is an oncology clinic. Onco means cancer. So oncogenes are genes that cause cancer. Let's see, what do the key terms say? An oncogene is a mutated version of a normal gene involved in the cell cycle can lead to cancer. So these are the bad versions. Proto-oncogenes are the normal versions. Let me read the key terms definition. A proto-oncogene is a normal gene involved in the cell cycle that, when mutated, becomes an oncogene. So this unrepaired DNA damage leads to proto-oncogenes becoming oncogenes. So we really don't want mutations, which means we really don't want unrepaired DNA damage. we can prevent cancer by making sure all of our damaged DNA is repaired properly. And in fact, mutations are prevented by things called tumor suppressor proteins or tumor suppressor genes. Again, every protein has a gene. Every house has a blueprint. So there are tumor suppressor genes that refers to the DNA that has the code for this. There are also tumor suppressor proteins. Those are the proteins that come from these genes. But either way, these are both used. Tumor suppressor genes and tumor suppressor proteins prevent mutations. How do they do that? Well, let's look at one of these in particular. This is tumor suppressor genes proteins. This is an incredibly important protein with a very humble name, P53, lowercase p even, P53. So what does P53 do? Well, here's a outline of this. P53 looks for damage, patrols the chromosomes looking for some sort of damage to the DNA. If it detects damage, it brings in DNA repair enzymes. There are many of these for different types of damage that can fix this damage. That's not all that P53 does. It also has a very important switch that it can pull one way or the other. If this damage is repaired successfully, P53 can give the green light on cell division and let everything go as normal. However, if this damage is too severe, or there's too much of it, or it just cannot be fixed, P53 also has its finger on the self-destruct button. And it can cause this cell, if it has damaged DNA that you can't fix, to destroy itself, which should look familiar. This is apoptosis from the previous chapter. So P53 can trigger this if the damage is too severe. So P53 locates damaged DNA, brings in repair proteins to fix it. If the repair is successful, cell cycle proceeds. If the damage is too severe, apoptosis. And so, I say my tests are not cumulative. I mentioned apoptosis in the previous chapter, which is part of the previous exam. I'm bringing it up again here, so I put it in the key terms again here. Apoptosis, programmed cell death. So, this sounds pretty good. It sounds like a pretty nice deal. Sounds like a pretty effective protein. If you can destroy cells before they accumulate mutations, then you'll never get mutations. You'll never get oncogenes coming out of proto-oncogenes. Fantastic. Nothing is perfect. This is what P53 is supposed to do. Nothing works perfectly in a cell. It is possible for DNA damage to sort of slip past the notice of P53. Even with P53 doing a fantastic job of all of this, it is still possible to get a mutation in, cyclin A's gene or any of these other cell cycle regulation proteins. It's still possible to get an oncogene. That by itself is actually not usually the end of the world. There are so many of these proteins involved here. There is a fair amount of redundancy in cell cycle regulation. Having one oncogene is not usually enough to cause really aggressive, deadly bad cancer. But again, this mutation could happen anywhere. It could happen in cyclin A, it could happen in any of these other things. The worst thing that could possibly happen is a mutation, unrepaired DNA damage, a mutation to slip past the notice of P53, but that mutation is in the gene for P53 itself. So if P53's gene becomes mutated, and this can happen, that means every single P53 protein that you build using those damaged instructions is not going to work properly. It's not going to be able to bring in DNA repair enzymes. It's not going to be able to trigger apoptosis as needed. If P53 goes down, everything is unleashed. You get way more unrepaired DNA damage, you get more mutations, you get more oncogenes. Again, one oncogene is not usually enough to cause cancer. You get 1, 2, 3, 4, 5. You get a whole bunch of oncogenes accumulating if P53 is not doing its job correctly. So if the P53 gene is mutated, it no longer encodes a functional protein. This allows cells with damaged DNA to divide willy-nilly, leading to even more mutations and even more oncogenes. So again, P53, incredibly important protein, but it's not immune to mutation. Here's an interesting statistic. In about 50% of all cancers that have been genetically studied, there's a mutation in P53 protein. So there are a few other tumor suppressor proteins and genes out there. P53 is a really big one. A lot of the time, this just sort of starts the ball rolling towards the creation of cancer if the gene for P53 is mutated. So that's an example of a tumor suppressor protein, what it's supposed to do, and sort of the worst possible scenario of how we can get lots of oncogenes and how we can get a cancer cell. Okay, that brings us to the end of chapter 10. We're going to, within this same recorded lecture, because that was kind of short so far, start up on chapter 11, which is actually still about cell division. It's just a slightly different type of cell division. So this has to do with sexual reproduction. So here's a slide from the very first day of class. Reproducing was something that all living things did. This was an example of asexual reproduction, but now we're going to focus on sexual reproduction. So virtually all, there's some exceptions, but virtually all multicellular eukaryotes, plants and animals, are capable of sexual reproduction. This is a big deal because sexual reproduction, as we will see, mixes things up. Asexual reproduction makes a clone of yourself. Binary fission, the stuff we saw in the last chapter, those made identical daughter cells. The big picture point of what we're going to see in this chapter is not to make identical cells. It's to have diverse offspring. It's to randomize things. It's to mix things up. Diversity is very valuable in organisms because you don't know what the next year is going to bring, what the next season is going to bring. If you have lots of offspring that have lots of different combinations of attributes, you're sort of hedging your bets and increasing the likelihood that one of those is going to have what it takes to survive whatever the world throws at them down the line. So again, this is sort of the big picture sales pitch on why all of this is needed, why we can't just do asexual reproduction for everything. Sexual reproduction mixes things up, creates diversity in offspring, and high diversity in offspring increases likelihood of survival. So, what is sexual reproduction? Well, let's break this down into the most exciting way possible, mathematically. So, at the end of the day, sexual reproduction is very simply sperm plus egg equals zygote. This is how it happens in animals. This is how it happens in plants. One cell plus other cell equals this cell. The zygote is this cell here. Now, a term that I introduced in the last chapter was the term diploid. I told you that virtually all of our cells are diploid. The abbreviation for this is 2N. That meant 2 copies of each chromosome, and we saw that in the slide on human chromosomes, 2 copies of each one of those. So if our sperm are diploid, 2N, and the egg is diploid, 2N, 2 plus 2 equals 4, that would make a zygote that is 4N? 4 copies of each chromosome? Do we double the chromosome number in our offspring? No, we don't actually do. Immediately crossing this out, we don't do this. this is the reason why I said virtually all of our cells are diploid. Yes, our skin cells are diploid, our muscle cells are diploid, our neurons are diploid. The exception is the eggs and sperm. These things are not diploid, they are 1N or haploid. That's because we need to generate a zygote that's 2N. We need to make offspring that is diploid, and the only way to add two numbers together and have it equal 2 is each of those numbers has to be 1. So sperm is haploid. These cells have one copy of each chromosome. Eggs are haploid. They have one copy of each chromosome, so that when they come together, the zygote is diploid, 2 copies of each chromosome. Now, the tricky part is we need a way to make these haploid cells. Everything we talked about in the last chapter, mitosis as part of the overall eukaryotic cell cycle, that made identical copies of cells. Here, we need to make haploid cells. No other cells in the body are haploid. We need a way to make haploid eggs and haploid sperm from diploid cells. The way that we are going to do this is through a process called meiosis. Some people say meiosis. That's another ligand-ligand thing. I say meiosis, but meiosis is fine. Meiosis is the process that's going to create these haploid cells. And this is done by special germ cells. So not everything is doing meiosis. Only germ cells within the testes or the ovaries are able to do this very special type of cell division. So yeah, here's a overview of the human sexual life cycle. You get a zygote, this single cell. It's going to divide. It's going to grow into an embryo, a fetus, a baby, an adult. All these cells are diploid. Then within the germline, we have meiosis to produce sperm or egg, as the case may be. These are haploid. The sperm is haploid. The egg is haploid. Here's the 1 + 1 = 2. Fertilization, we're back to diploid, and it's, back to the rest of this cycle. So meiosis is the process here, and germ cells are the cells that do meiosis. Again, not everything does this. So if we want to talk about the process of meiosis, I talked about how much I liked this particular figure in the last chapter, so I'm actually just going to modify this. So this was mitosis as part of an overall eukaryotic cell cycle. Really, we just cross these ones out and replace them with this. So this is what happens specifically in eukaryotic germ cells. We have G1, S, G2, and then we have meiosis 1, cytokinesis, meiosis 2, then another cytokinesis. And so obviously this is going to be a 2-step process. It's not just one division, it's 2 sets of divisions, 2 cytokinesises. This is going to result in 4 cells by the time we're done with this. It's 2 rounds of cell division. So we'll get some visuals on this soon enough. But yeah, I liked modifying this familiar figure. This is how it happens when a cell is doing meiosis, when a germ cell is doing meiosis. So, all right, eukaryotic germ cell cycle. We got interphase. Not going to say anything more about that. We talked about G1S and G2 in the last chapter. I'm not going to rehash any of that again. It's the same interphase I talked about then. Copy and paste all that in. All right, next we have meiosis 1. So meiosis 1 has the same PPMAT that mitosis had. So in some ways this is convenient that it has so much in common with mitosis, but in other ways it can sometimes be confusing because it has so much in common with mitosis. I am going to do my best to highlight the similarities and the differences between the PPMAT of meiosis 1 and the PPMAT of mitosis from the last chapter. The way that we distinguish these in text is with the Roman numeral 1. So if we're talking about prophase 1, that means we're talking about the prophase of meiosis 1. if you see and so on my multiple choice test questions, you have to read these questions carefully. I'm not trying to trick anyone ever. I just want you to read them carefully. If the question says metaphase 1, it means I'm talking about the metaphase of meiosis 1. And spoilers for later on, if I say metaphase 2, that means I'm going to be talking about the metaphase of meiosis 2. And if I just say metaphase flat with no additional Roman numerals, that means I'm talking about the metaphase from the last chapter, the metaphase of mitosis. So again, not trying to get confusing, but the Roman numeral is how we distinguish these from mitosis. So, all right, let's take a look at this process. We got prophase, metaphase is not pictured here, I think just to save space on this figure. Metaphase, anaphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and then finally cytokinesis. So let's start in on prophase 1. So prophase 1, we are going to have the chromosomes condense. The nucleus, Golgi, and ER break down. Okay, this should sound familiar, but I guess that's good. Prophase 1 is very similar to prophase from mitosis. Nucleus, Golgi, ER break down, chromosomes condense. So far so good. Has a lot in common with mitosis. We are going to see the chromosomes. Oh, okay, so this is actually a little different now that we look at it. Back in mitosis, we had sister chromatids appearing at this point. We had these 2 chromosomes partnered up, but if we look closely at this, that's not 2, that's 4 total chromosomes. If we want to zoom in on this so we can get a better look at this, yeah, that's 1, 2, 3, 4 total chromosomes paired up together. This structure of 4 is, this is new to meiosis 1. This structure of 4 is called a tetrad. And tetra means 4, so there are 4 total chromosomes here. This consists of 2 pairs of sister chromatids. So these 2 red ones here, these are the sister chromatids. The 2 blue ones back here, these are sister chromatids. So what are the red versus blue? Why are these different colors? Well, they're labeled here, the red versus blue are homologous chromosomes. So, okay, let me write this down, then I'll explain what homologous chromosomes are, because this is a very important term. We've got prophase 1 still. We're going to form tetrads, which the key terms define as 2 duplicated homologous chromosomes, parentheses, 4 chromatids, and tetra means 4, bound together during prophase 1. So this set of 4. Synapsis is the name of this process. So synapsis occurs during prophase 1, and it leads to the formation of these tetrads. So, okay, what are homologous chromosomes? Well, homologous chromosomes are not identical. Sister chromatids are identical. They're exactly the same. Homologous chromosomes have the same genes, but their exact sequences are a little bit different. This needs an analogy. These are sister chairs. These 2 chairs are exactly the same. No differences at all. Just like these 2 sister chromatids, they just went through DNA replication. These are exactly the same. Sister chairs are exactly the same. Sister chromatids are exactly the same. Homologous chairs are the same piece of furniture, but they are not exactly the same. These are both chairs. They are the same thing. They're the same type of thing. But, legs are connected in a slightly different way here. The slats run vertical versus horizontal. I think this one's maybe a little bit wider than this one. They both do the same job. They both fulfill the same function of you sit on this thing. But they are obviously not exactly the same. That's what homologous means. They have the same genes. They have the same basic structural foundations of 4 legs, a seat, and a back. Homologous chromosomes are the same type of thing, but their exact sequences are a little bit different. In these textbook figures, that's what the red and the blue, these different colors, are always trying to make clear. If it's the same color and the same size and they're together, they're exactly the same. Sister chromatids, sister chairs. The red and blue is meant to show that there are differences here. The red and the blue are homologous chromosomes, the same genes, but different exact sequences. And again, this is a good summary of what this is. Homologous chromosomes have the same genes as one another, but the exact sequences are a little bit different. So this tetrad has 2 sets of homologous chromosomes and their sister chromatids. So 2 sets of sister chromatids, 4 of these things total. Okay, I spent so much time on this because this is one of the most important things, one of the most important differences between this and mitosis. So, why? what was the purpose of doing this? What was the purpose of forming this tetrad? We didn't do it in mitosis. Why are we doing this? We go through synapsis and we form these tetrads in order to be able to do something called crossover, or crossover. There's another term for it I'm not going to use. Crossover is exactly what it sounds like. It's when these homologous chromosomes swap a little bit of information with one another. If we were looking at chairs, I would rip off this front left leg, swap it with this front left leg. You're left with 2 still completely functional chairs, but they've swapped a couple of parts with one another. And the purpose of this is to make slightly different chromosomes. You think about parents and offspring. This chromosome is coming from one parent. This chromosome is coming from the other parent. You're trying to make offspring of your own. If you're doing crossover, here's a slightly different chromosome. It's not the same as any chromosome you have. It's not the same as any of your parents' chromosomes. You're mixing things up. You're increasing genetic variation. And every time your body goes to make eggs or sperm, it's doing this crossover in a slightly different way. It's not always down here at this exact position. So this, again, increases genetic variation. The whole point of this is to not have the same offspring twice. We're not having millions of offspring, but there are other organisms out there, especially big trees, that can make millions of sperm cells, millions of egg cells, and you want to have as much variation as possible. So this synapsis allows for crossover. The purpose of crossover, which, I'm sorry, I think I forgot to read the key terms definition of crossover. The definition of crossover from the key terms is the exchange of genetic material between homologous chromatids, resulting in chromosomes that incorporate genes from both parents. Allows for crossover, and the point of this is to increase genetic variation. Okay, all of this was happening during Prophase I. Next, we've got Prometaphase I. This one's easy. Microtubules attach to the kinetochores of these tetrads. Pretty similar to the last chapter, microtubules attach. But again, it's tetrads they're attaching to, not sister chromatids. Got this set of 4 here during Prometaphase I. Okay. After Prometaphase I comes Metaphase I. And, very similar to last chapter. Everything's lining up in the middle. But there's an important detail about this lining up in the middle. So, Metaphase I, these tetrads all line up in the center of the cell. But importantly, it matters which way they are facing. So back in mitosis metaphase, it didn't matter whether this chromatid went to the left or whether this chromatid went to the left, because these 2 chromatids are exactly the same. The 2 sides are going to get exactly the same stuff. But here, it does matter how they are facing. We can picture, sorry, this one's horizontal instead of vertical, this figure, but we can imagine what's going to happen next. They're going to get pulled apart from one another, and it looks like the daughter cell up here is going to get red for this chromosome, and the daughter cell down here is going to get blue for these chromosomes. So it does matter which way they line up, which way they are facing. So how do we, make sure that each daughter cell gets about the same amount of red and blue? How do we ensure an equal distribution of these different homologs? Well, the answer is we don't. It's completely random which way they are facing. So let's compare these chromosomes once again to pieces of furniture. So yeah, you have two different chairs. You have two different tables. Again, these are all homologs. You have two different nightstands. You have two different chest of drawers. This is where I got bored of Google image searching different furnitures. Suffice it to say, in a human with 23 different chromosomes, this would be a pretty crowded room indeed. But yeah, these line up in such a way that you're going to make an egg or sperm cell with this chair, this table, this nightstand, this chest of drawers, and a different egg or sperm cell with this chair, this table, this nightstand, this chest of drawers. Or, you could do this with this combination of furniture and another cell with this combination of furniture. Or it could be this combination and this combination. Any of these are possible. Here is a simple organism with only 3 chromosomes, showing all the possible combinations. You have blue, 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 red, 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 or red, red, blue, and blue, blue, red, and blue, blue, red. And you see what I mean? 8 possible combinations if there are 3 chromosomes. I did the math for you. In an organism like ourselves, with 23 different chromosomes, there are over 8 million possible combinations for how these homologs can be sorted. That means you could go through the process of making egg or sperm, as the case may be, 8 million times and not make the same cell twice. The whole purpose of this is to increase genetic variation. So in metaphase I, these tetrads align at the center of the cell. Which way they're facing, their orientation is random. So different daughter cells will receive different homologs. They're not going to be the same every time. The purpose of this is to increase genetic variation. Okay, that was PPMAT. After metaphase I, we come to anaphase I. And the tetrads are pulled apart from one another. Importantly, the sister chromatids stay together. They'll get pulled apart in the next one. But yeah, the tetrad is broken apart and the sister chromatids stay together, pulled to opposite sides during anaphase I. Yep. Tetrads pull apart, sister chromatids go to the same side. Okay. And here, if we want to zoom in on this, this was prometaphase where the microtubules attached, and there is anaphase I where the sister chromatids sticking together, but the tetrad is broken apart. And finally, telophase I, followed afterwards by cytokinesis. Very similar to last chapter. Telophase I, chromosomes decondense, nucleus Golgi ER are going to reform X2, and then cytokinesis is going to pinch that cell into 2. So at this point, we have finished meiosis I and the first of our 2 cytokinesis. The question is, what kind of cells are these daughter cells? Are these daughter cells haploid or diploid? Don't answer yet because this is a little tricky. At first count, you may look at this and say, oh, this daughter cell has 1, 2 chromosome, 1, 2, 1, 2. This looks like a diploid cell. Technically, diploid means you have 2 different things. You have 2 different chairs. you have two different nightstands. You have two different tables. This is our default cells. We have two different versions of everything. These cells here, at the end of meiosis I in the first cytokinesis, are not actually diploid. These two chromosomes are the same. A little bit of difference because of crossover, but these are the same chromosome. These are the same chromosome. These are the same chromosome. These daughter cells are, and this is an awkward phrase, but it is what it is, these cells are haploid with replicated furniture, or haploid with replicated DNA. So yeah, this is not a diploid room. This is a haploid room with replicated furniture. Two of the same chair, two of the same table, two of the same nightstand. And that's what these cells are at this point. So after this first cytokinesis, we have two cells. They are haploid with replicated DNA cells. They're not diploid cells. They don't have two different things. They have two of the same thing. Haploid with replicated DNA. Okay. Whew. Back to this. We did meiosis I. We did cytokinesis. Now it's time for meiosis II. Importantly, there is no interphase between meiosis I and meiosis II. You don't have another cell growth. You don't have another DNA replication. You don't have another G2. After cytokinesis, you go to meiosis II. So, what happens in meiosis II? Well, got another figure to show this. So we got prophase II, prometaphase II, not pictured here, metaphase II, anaphase II, telophase II, cytokinesis. All right, so what's going on here? Well, prophase II. It looks like the ER, Golgi nucleus are breaking down. The chromosomes are condensing, becoming visibly distinct. This is exactly the same as it was in mitosis. Prometaphase II, again, it's not pictured here, but the microtubules attach to the sister chromatids. No tetrads here. No crossover is happening here. No synapses here. This prophase II, again, I'm sorry it's not pictured here, is exactly the same as the prometaphase from the last chapter. Metaphase II. Sister chromatids line up in the center. Okay, still no tetrads, no crossover. These sister chromatids lining up exactly the same as it was in mitosis. Anaphase II. The sister chromatids are pulled apart exactly like it was in mitosis. And then telophase II and cytokinesis, chromosomes decondense, nucleus, ER, Golgi, all the same stuff from mitosis. So although meiosis I had, several important differences comparing it to mitosis, meiosis II is actually the same. Everything that I would write down about prophase II is exactly what I wrote down for prophase in the previous chapter. Everything for prometaphase II is the same. You can, if you're making your own notes and you want to do this, you can copy and paste this in if you want to. I'm going to save us all some time and just say each one of these phases is the same as the phase from mitosis. There are no notable differences between prophase II, prometaphase II, metaphase II, anaphase II, and telophase II, and prophase, prometaphase, metaphase, anaphase, telophase. Exact same descriptions I gave in the last chapter. So by the time we finish with this second cytokinesis happening right after telophase II, we are finally left with 4 haploid, truly haploid, not haploid with replicated DNA. We are truly left with 4 haploid cells. So, let's go back to furniture once again. We started with a diploid room, 2 copies of each chromosome. We had interphase, which means we had DNA replication as part of interphase. We created a diploid room with replicated furniture. It's starting to look a little messy, but this is what happens during interphase. We replicated everything. After meiosis I and that first cytokinesis, that left us with a haploid with replicated furniture room. We only had one of each type of furniture, but we had 2 copies of it, haploid with replicated furniture. And by the time we have finished Meiosis II and the cytokinesis that comes after that, we have a truly haploid room. One single copy of each piece of furniture. Sorry if this furniture analogy is getting a little tedious, but it's a good way, I think, to describe what's going on here. By the time we finish this second cytokinesis, we now have 4 haploid cells. If you don't appreciate furniture, if you don't, that's fine. Here's an overview of meiosis compared, side by side, with mitosis. I find this very useful as a visual. Hey, if you can draw this yourself, if you could, and I'm not a great artist. Even if you're not a great artist, you could draw a cell. It's just a circle. You could draw a chromosome. It's just a sausage. You could get 2 different colored markers or colored pencils or pens, whatever. If you could draw this out yourself, you'll really get a better appreciation for what's happening along the way. Mitosis was interphase, prophase, prometaphase, metaphase, anaphase, telophase, cytokinesis. Done. Meiosis was interphase, prophase I, prometaphase I, metaphase I, anaphase I, telophase I, cytokinesis. Each of these cells goes through prophase II, prometaphase II, metaphase II, anaphase II, telophase II, cytokinesis, ending up with a total of 4 cells, all of which are haploid. I don't have anything to write down from this slide. I think it's a fantastic visual overview of this entire chapter and kind of the last chapter as well, but I have no new information to add. This is just a great visual resource for you. And here's another great visual resource for you, just sort of breaking down Meiosis I, and here's one breaking down Meiosis II. Again, nothing new for me to write. I just want to give you as many resources as possible to give you something visual to look at as you're studying. So that brings us to the end of this chapter on sexual reproduction and meiosis, and this is the end of recorded lecture 3-2.